A very good morning to all of you. Now I will start our fifth online lecture on plant molecular biology techniques and plant molecular biology. Now, in this class, I will mainly highlight about the different techniques of plant transformation and the very useful techniques like agrobacterium mediated transformation and how we can transfer our clone gene into the plants through agrobacterium, through ballistic method. So different methods of plant transformation and what are the advantage or the demerits of this system and how we can do that. Now, and the first first slide here depicts that we need to transform plants. Why we need to transform plants? This is the question. Because simply you can say that we need to overexpress or we want to develop some character within a plant or to make a transgenic plant which will express this character or which will carry an improved trait. These are for the application. Definitely, this is one of the side of transformation. But another side will be there in which I can show you that this is a basic requirement for plant molecular biologist because if we want to characterize the gene or if we want to know the function of that gene or if we want to validate the function of that gene of a particular gene, then we have to express or we have to overexpress it or we have to silent it, whatever it may be, for any kind of techniques or for any kind of protocol to be adopted. Plant transformation or the genetic transformation of plants is very, very essential. Otherwise, we couldn't able we are not able to develop a transgenic plant or the transformed plant where we can represent that yes, this gene is working like this or this gene has this type of function. We can't confirm the function of the gene. So this is very important to identify the gene function. Suppose, for example, a gene is associated with a particular type of stress resistance in plants. It may be the biotic stress, it may be the abiotic stress in any kind of. So I know I have the preliminary knowledge or the preliminary data that a gene is related with drought tolerance or biotic stress tolerance. But if I want to confirm this function that yes, these gene, if we overexpress in the plant, then the plant become drought tolerant, or if we can mutate, or if we can make a knockout mutate copy in the plant for this gene, then the plant become susceptible to that particular kind of stress or drought stress. So to prepare to develop this kind of overexpression plant or the silent plant or any kind of mutated plant, we need to transform this plant. First, we need to identify the gene, we have to clone the gene, and then this gene should be transferred from a vector to the plants, and this gene should be integrated in the plant genome. So the successful introduction and as well as the integration of the exogenous DNA into the plant cells can give rise to the transgenic plant as this is an essential system and the plant molecular biologists are randomly use this system to validate the gene expression in the lab. So it is very important to identify the gene function or to confirm the gene function and as well as parallelly if we can introduce the new trait or to eliminate the harmful character that will definitely produce the transgenic plant or the GM crop or GM plants that will ultimately uh, help the human society or the human welfare. So this is the application of this transgenic technology or the gene transfer technology. Now, there are some methods or which we can adapt for gene transfer or for plant transformation. First, it is very natural system. Through the agrobacterium mediated gene transfer method, we can transfer our gene of interest into the plants. This is very efficient, but the only limitation is only a selected group of plants, dicot plant in most of the cases, and some monocot plant, we can use it for transformation. Now, the next part with the direct or vector-less DNA transfer. So that is by physical method, electroporation, microprojectile, that is a particle bombardment. I will highlight also this part, the particle bombardment mediated gene transfer. Microinjection, there I can inject the cell or our gene of interest into the particular type of cell like protoplasm. Then the liposome fusion, we can fuse the protoplasm with a particular type of liposome, that is a lipid membrane covered substance carrying our gene of interest. Through liposome mediated fusion, I can develop a transgenic plant mainly from the protoplast. Silicon carbide fibers, that also requires the regenerable cell suspension. Suspension culture of the liquid cell culture is very, very important for this. So these are some physical methods, and some chemical methods are also there. There is a PEG mediated or polyethylene glycol mediated. That is also mainly confined to protoplast. Fertile plants, regeneration of fertile plants is frequently problematical in this case, the PEG mediated generation. And another is a DAE, dextran mediated generation of the transgenic plant or the plant transformation. But here, in this presentation, we will mainly focus first on the agrobacterium mediated gene transfer method, and second, on the physical method, that is the biolistic. Now, first, agrobacterium mediated gene transfer in plants. Now, what happens when in the natural system, agrobacterium is associated with the plant and it can transfer its DNA part? Scientists are utilizing this system. They didn't develop that the association or the how agrobacterium can infect. This system is already known. So how agrobacterium infect the plant and it can introduce its DNA region into the plants. This is already established, and this is the natural system. First, what happened during infection? The association between agrobacterium and the plant is occurred when there is need of agrobacterium to develop some crown gall disease. Agrobacterium tumefaciens cause a crown gall disease in plant, in most of the dicot plant. And Agrobacterium rhizogens, this is another strain. It can infect the root system and develop the hairy root culture or hairy root system. So, the crown gall disease, the tumor inducing plasmid is there within the agrobacterium, that is called TI plasmid. So, these tumor inducing plasmid, before the crown gall disease or before production of the tumor, what happens? The agrobacterium can infect the plant depending on the plant signal. It can recognize the plant signal. And what is the plant signal? There are different types of acid or phenolics or sugar moieties. It varies plant to plant or species to species. If I consider the phenolics, that can activate the agrobacterium gene for the association. The gene may be the wheat genes. Phenolics mainly activate the wheat genes, or some sugar molecules that can also activate some other genes like CHBE genes or CHB genes, that is a chitin-related genes. And some acids are secreted by the plants, which can also recognize by the wheat G protein in agrobacterium. So this this signaling, the chemotactic movement or the signaling is very, very important for the association between agrobacterium and the plant. Now, once this association has been formed, then what happens? The downstream signaling cascade within the agrobacterium is now open. Now, after after recognition by VIR-A, downstream VIR-G got phosphorylated, and this is a master regulator. Once the VIR-G got phosphorylated, then it can induce the expression of other VIR genes which are present in the TI plasmid. And once this VIR gene sequentially expressed, then one of the VIR gene or VIR gene product, that is a VD, VD2 mainly, VD1 and VD2 is there. VD2 or VD1 mainly. VD1 possesses an endonuclease activity. And this winter VD1, it can cut the tDNA region at the right border site. This is the tDNA. You can look into it. That is the tDNA region. And this tDNA possesses the two repeat border. One is right border, one is left border. And towards the repeat border, 25 nucleotide repeat border sequence, VD1, it can cut the tDNA at a particular sequence, and then VD2, just capping that pi prime end. Sorry, that it acts as a capping like molecule to protect this tDNA from the degradation into the plant cell. Then, after capped, the tDNA binds with the different VIR-E2 protein. The next protein, VIR protein, is the VIR-E2. And once the single-stranded tDNA, not the double-stranded, single-stranded tDNA, bind it with the VIR-E2 and capped by VD2, then is channelized into the plant cell through a channel, and this channel is made up of like different protein along with the VIR-B protein. So VIR-B protein facilitates the channel movement or the preparation of that particular channel through which the tDNA is transferred. So tDNA is transferred along with the T complex, and this T complex is nothing but the VD2 protein binding, binds with VD2 protein bind, and the VIR-E2. These VIR factors, virulent factors, this VIR-E2 bind to the tDNA. Next, VIR-F. This is also associated with the VD, and it can direct the tDNA into the nucleus, from the cytoplasm to the nucleus. And once it is incorporated into the nucleus, then it will be integrated in the genome, depending on the particular site-specific recombination. So, this is the mechanism by which tDNA is transferred from agrobacterium to the plant nucleus, and it got integrated into the genomic DNA of the plant, and then it will produce the cytokinin, auxin. These genes are present within the tDNA region. So only the tDNA region is transferred, and within this tDNA region, there is auxin and the cytokinin biosynthesis gene, and another is the opine, that metabolic product. This is another special kind of metabolic product that is required by agrobacterium cells. So for the growth and the development of the agrobacterium. So these opine, these auxin, these cytokinin, they are present within the tDNA, and once the tDNA got integrated, they resume their function, and auxin can overproduction of the auxin can induce the root formation. The cytokinin, higher production of the cytokinin, it induces the crown gall formation of the shoot multiplication. So this is the system. So now, for our case, scientists, what they are doing, they want to incorporate their gene within the tDNA so that it will be transferred directly to the plant genome. Because gene transfer means I want to introduce my gene or my expression cassette within the plant genome. Not only this, successful integration. So it is only possible if my expression cassette or the gene of interest is placed within the TDN. So first, we should keep in mind that for the successful introduction or the integration of the tDNA, we need left border, right border site. There is that can flank the tDNA, the direct orientation. Okay. Next, what are the genes required? Read genes. Read gene plays an important role. So read genes should be located within the TI plasmid or the RI plasmid. Now, other chromosomal genes definitely should be possessed within the Agrobacterium that involve in bacterial exopolysaccharide synthesis or maturation and secretion that ultimately actually helps to recognize the plant signal. So this system should be present within the Agrobacterium strain where we will introduce our expression cassette or the recombinant vector. So that scientists, they have developed some vector based on this tumor-inducing plasmid. We only focus on the tDNA region. Others are there. Rest will be there. Virulence region, definitely for Agrobacterium maintenance of this plasmid, Odyssey region, all should be there. Opine catabolism genes should be there. But we have to focus on the tDNA region. Right border, left border, intact. Now, these auxin production gene and the cytokinin production gene, these two genes are required for the plants for the development of the crown gall or hairy root system. But it should not be our criteria because we want to express, we want to overexpress, or we want to express our gene. So that means our expression cassette should be there. So instead of cytokinin or auxin biosynthesis gene, if we can place our expression cassette, then it will be better. It can be adapted. Now, what scientists did, they just removed auxin and cytokinin genes or that part from this TI plasmid. And once removed, this plasmid is called disarmed plasmid, disarmed vector, disarmed. So, armed, there is cytokinin genes or the auxin genes, they are removed. Now, once removed, then we can incorporate our gene, our expression cassette in place of auxin biosynthesis gene. Our expression cassette means any gene, the gene of interest, before that, towards the 5' end or the upstream of the gene, there should be the promoter, and towards the 3' end of our gene of interest, there should be the terminator. So, or the gene might fuse with the GFP or GAS, anything. So, anyways, this expression cassette will be placed here. But not only this expression cassette, but another necessary thing is the another expression cassette, which is mainly required for the plant system. Why? Mainly required for the selection, for the recombinant plant selection. Recombinant plant means, suppose when we transform the cloned or the vector or recombinant vector into the bacteria, that bacteria harboring that recombinant vector will be selected against some antibiotic selection. So here, how we can identify that which plant is transformed or not, or the transforming plant, on the basis of the selectable marker gene. In case of the plant, we can use the several genes or the proteins like hygromycin, also the kanamycin is the antibiotic. These hygromycin or kanamycin, they will select the recombinant or the transformed plant. How? Hygromycin phosphotransferase, it can cleave, it can degrade the hygromycin. So the vector or this tDNA, if it carries that HPT or hygromycin phosphotransferase or NPT neomycin phosphotransferase, then if this gene is expressed in the plant system, the transgenic plant or the transformed plant, then the HPT will be expressed, and that HPT can degrade or can break the hygromycin and give the plant place against this plant. So on the basis of that, we can select. So this is called the selectable marker. So hygromycin or NPT, it should be placed under the control of a promoter. And that promoter, in most of the cases, we choose the constitutive promoter because it can express in any tissues and just for the selection. So on the basis of the expression or on the mainly on the basis of the antibiotic selection, we can select those transformed plants. So that cassette also should be here. That cassette also should be here. The promoter, selectable marker, and the terminator. Again, our cassette, the gene of interest cassette. That is the promoter, gene of interest, and the terminator. So two cassette should be present within the tDNA region, and as well as the opine metabolism or the opine biosynthesis gene. Now, so here, scientists have developed several binary vectors where the selectable marker cassette, and as well as the gene of interest cassette is there. Now, look at the picture that no read genes in this binary vector. So how tDNA will be transferred from this TI plasmid or binary vector to the plants? Now, in case of the binary vector, or scientists have developed two different types of systems, one by binary vector and one by co-integrated vector. In binary vectors, the examples of the binary vectors are so many, like PKMBA 1304, PBI 121, PGAT, PGB2B vector. These are all binary vectors. I think you have heard some of them. And what happened? In the Agrobacterium strain, which poses this binary vector, this is the binary vector based upon the TI plasmid. Mainly binary vector, it carries the tDNA region where my gene of interest and the selectable marker cassette will be there. And another vector system, another vector should be present, which carries only the V gene, no tDNA region. This vector is called a helper plasmid. So the binary system, that Agrobacterium strain will carry the tDNA containing plasmid or the recombinant vector, the tDNA vector, and as well as the helper plasmid. So this system or that tDNA carrying vector is called the binary vector, because with the help of the other vector, binary, that is within the Agrobacterium system, there is another system, there is another plasmid with the help of the other vector, or that possesses the mainly the V genes. With the help of that V genes, the tDNA will be transferred from the Agrobacterium towards the plants when the plants are infected or co-cultured with that Agrobacterium strain. But what happens in co-integrated vector? In case of the co-integrated vector, no helper plasmid is required. The single plasmid or single vector can carry the V genes as well as the gene of interest, my gene of interest expression case. So this can be done by a simple conjugation between E. coli and Agrobacterium. So this is co-integrated vector and this is binary vector. So by recombination, the single vector, we can produce the co-integrated vector. This is mainly by the recombination, and a single vector that contains the V genes and the tDNA within the same Agrobacterium strain. So this is important. Now, these are several examples of the PKMB binary vector, like PKMB 1304. This is the tDNA cassette. Now look at this picture that here the selectable marker cassette and placed towards the left border side, and here the gene of expression cassette like GAS-A, GFP, 35S promoter. You can replace the GFP or GAS from here with your gene expression, with your GOI or gene of interest, or you can clone your gene of interest here. And this is the terminator. So this is one cassette and this is another cassette. These are placed within the left border, right border region. Next, this is PBI 121, where NPT2, that is a neomycin phosphotransferase, is used as selectable marker. This is the PIGAD, where BASTA or BASTA gene, another that BART gene that also does phosphoinotrypsin. Phosphoinotrypsin or BASTA. It also confers some BASTA resistant for the development of the BASTA or phosphoinotrypsin resistant plant. This is also behave as a selectable marker. So you can clone your gene here, polylinker region. So this is placed towards the right border site and the selectable marker gene towards the left border site. So these are the different types of vectors. Now the important question that is there any rule to to place my gene of interest towards the right border or towards the left border, my gene of cassette, my gene of interest cassette? No, there is not a mandatory rule, but in most of the cases, you can find it out that towards the left border side, my expression cassette will be there, because the different companies have developed this binary vector. The one of the reason of this orientation is that when the chopping of the RB site, when the chopping of the tDNA, it mainly occurred at the right border site first. So, and so which will be the first site to be incorporated into the plant? RB site. That means the right border site. So, if we place our gene of interest cassette towards the right border site, it will be first introduced. And definitely the left border will be introduced, but it will be introduced later. So, in case of if there is a chance of incomplete introduction or incomplete integration, then my gene of interest can be placed or it can be integrated there. Maybe the selectable marker is not integrated, but my gene of cassette will be integrated. So for this, although it will create some problem during selection, but if a part of the selectable marker gene is also introduced, then I can at least select those plants on the basis of the transient expression of the selectable marker. So for these, some companies have developed a system where, in case of the PKMBR 1304, in case of the PIGAD, you can find that the gene expression cassette will be placed towards the right border region. So it is better if you can place or if you choose a vector where towards the right border side, your gene of interest cassette will be placed. Now, after development of the recombinant vector, so we need not to worry about the construction of the vector, because we will simply clone the gene in the TA vector depending on the depending on the T&A or TA system, or depending on the restriction enzyme based digestion ligation system. We can clone the gene from the plant or from anywhere to a particular vector. But from this vector, we have to construct a binary vector, and we need not to worry about the whole construction, because we will subclone the gene from a TA vector to the binary vector or the destination vector when we want to transform the plant. So other part like selectable marker, gene expression cassette, or the promoter region, all are there. All are present in the binary vector. Just we will clone our gene of interest into the binary vector, only the gene of interest. Or if you want to replace the promoter with some specific promoter like tissue specific promoter or inducible promoter, that you can do that. But this is our criteria. Scientists or the companies, they have developed the system, the vector system, binary vector system, which we will just use for the cloning of our gene. Now, what happened? So, suppose we have developed a recombinant binary vector where my gene of interest is there. So you have already learned about the different cloning techniques and procedure. Through this, we are able to clone, and we have developed a recombinant vector. That recombinant vector, next, what we will do, we will transform it into the agrobacterium strain, the empty agrobacterium strain. There are so many strains. Empty agrobacterium strain where the binary vector is introduced. The empty means that agrobacterium strain will carry a helper plasmid. That helper plasmid poses a virulence gene. So one plasmid should be there. That is the helper plasmid. Empty for our binary vector. Okay. Now, once I will introduce this binary vector towards the agrobacterium by simply transformation, bacterial transformation system. Then we'll select the agrobacterium on the basis of the recombinant agrobacterium strain on the basis of the antibiotic. One antibiotic for my gene of interest or my vector, and one for the agrobacterium, another agrobacterium system. So rifampicin in most of the cases for agrobacterium it is used, and one antibiotic that is present in my binary vector. It may be the kanamycin, it may be the ampicillin, whatever it may be. Just like E. coli system. Next, what we will do, we will check that whether recombinant vector is introduced within the agrobacterium or not. So through colony PCR or through restriction in enzyme digestion, we can check that yes, my gene of interest is there. And then once we confirm that yes, this agrobacterium strain will possess the binary vector, my gene of interest cassette, or the my binary vector, then the explants, we require the explants to co-culture with this agrobacterium strain. Explants means the plant part which are able to produce a total system or total plantlet. That means which are able to produce a complete, a single plantlet. Okay, that means this is the totipotence, the nature of the plant, regenerated plant from a explant. It may be embryo, leaf pod, cotyledonary leaves, and as well as the calli. For indirect organogenesis, we need the calli, where direct organogenesis from any explant like embryo, leaves are not possible. So these explants, it may be the calli, it may be the embryo, it may be the leaves, are co-cultured with the agrobacterium. That agrobacterium forwards the recombinant binary vector. Then there are some techniques through which we can co-culture, and then we wash this explant after incubation period, after incubating it in a dark condition, then in the light condition. Then after 2 to 3 days, we will wash the calli or the explant with the proper antibiotics media or with the proper antibiotic solution to remove the excess amount of the agrobacterium, because excess or overgrowth of agrobacterium can hamper the regeneration of the explant. Next, the regeneration or the organogenesis of the transformed plants on the plant selection media, or before the regeneration, we can also select the calli, the transformed calli or the transformed explants on the selection media like hygromycin containing media, like kanamycin containing media. So after the selection, the selectable calli, the selected calli or the selected explant will be transformed into the regeneration media for shoot regeneration. Shoot regeneration is very, very crucial. Once the shoot regenerated from the selected calli, then the shoot will be placed on the rooting medium for the root development. Once the root has been developed, so now we will get a complete plant. So each plantlet will be transferred to the soil or other things for the hardening. Then, so this is, look at this picture. So this is shoot, then root will get the total plant. And so multiple plants can be generated from the multiple explants or the calli. So suppose 50, 100 plants, transformed plants are generated. Then I will confirm that yes, all these plants carry my gene of interest or the TDNA region or not. To confirm it, I will check through PCR, genomic DNA PCR with my gene-specific primers. So this is the basic principle and this is the basic protocol of agrobacterium-mediated transformation. Now, another transformation system. This is a particle bombardment physical. In case of the particle bombardment, we just coat the gold particle or the tungsten particle with our DNA molecule. Here, we can consider the vector, tDNA vector, binary vector, or the tDNA, only the tDNA part, or simply any kind of vector, because it doesn't require any agrobacterium strain. So it doesn't require the virulence gene at all. So any kind of plasmid or any gene construct which carries my gene of interest or gene of interest expression cassette, that is sufficient for this particle bombardment method. In particle bombardment method, there are some macrocarrier, microcarrier on the filter-like thing, and on this filter or the macrocarrier, we just coat this DNA or the plasmid with the gold coated or the tungsten coated that DNA. That tungsten coated DNA now will be placed here. Look at this. This is the ballistic or particle gun system. Placed it here. And the plate where the explants, these are the several explants, like 100, 200 explants. These, that particular explant or this plate are placed here. And then this is the pressure system that through helium gas or the pressure is like accelerated to 1100 or 1300 psi. And then we could press a switch automatically and the gold particle coated DNA will be bombarded directly into this plate where the explants are there. Just it will spread it over the explants with a force. And due to this penetrating force, the DNA will penetrate the embryo tissues or the explant tissues and just integrate it with the nuclear DNA. Not only the nuclear DNA, it can integrate with the plastidial DNA. And then we will select it, those introduced or those transformed explant on the selection media. Those which are growing on the selection media, those grown calli or those grown explants will be placed on the regeneration medium, just simple like as before. And then the regenerated plants or the regenerated seedlings will be transformed to the rooting media and then next grown in the greenhouse condition. Because any transgenic plants, we should grow in the greenhouse condition, not in the open air. So these are the basic protocol. Now, which method we will choose, agrobacterium or bialistic? Both agrobacterium and bialistic has merit and demerit. What are the merits of agrobacterium? That it is only required for the nucleus transformation, and from the agrobacterium, the gene is transferred or the gene of interest is transferred to the plant. That plant can carry this transgene in a very low copy number. It's a very low copy. So 1, 2, 3. And here I will mention that low copy is the desirable. Mainly the desirable criteria is single copy. If we want to introduce my gene of interest into the plant, the single copy trait or single copy carrying gene or single copy gene is essential. So we have to select, we have to choose those plants which will carry the single copy. If not, then we can go for the maximum 2 to 3 copies, not more than that, because the more copies, that means more genetic load, more other redundancy factor may possess a negative role towards the for the transgenic plant. Not only this, to getting a homozygous plant, because homozygous plant is very, very important for the stable trait development or for the generation studies. So for getting a homozygous plant, we need time, not only this, and it is quite difficult to analyze that which generation contains and which plants contains the homozygous line, which line contains the which independent event, which independent lines contains the homozygous copies or not. So if 2, 3, 4 copies or 5 copies, then the genetic load is more, as well as it poses some ambiguity in to getting to find it the homozygous line. So always the desirable is a low copy number. So and this tDNA can integrate at specific area of the host genome. But in case of the bialistic, this is not in our hand. We just bombarded it with the DNA coated, gold coated DNA particles. But the smaller fragment can integrate it randomly in anywhere in the host genome. This may produce a redundancy factor that if it integrates in the gene sequence or exonic region, exon part of any other gene, that can hamper the function of that particular gene. Suppose A gene is my gene of interest and A gene is introduced into the part of the B gene or the C gene, exonic region, then it will hamper. So it will produce some abnormal feature and then the transgenic plant. Or it may be introduced into the intronic part. Ultimately, my gene of interest in the plant, in the transplant plant, become silent. So there are some chances because this is not in our hand, this is randomly occurred. In bialistic and high copy number is commonly found because the insertion is random. In case of the agrobacterium, for the single copy number inherited to the later generation as a stable trait, or to getting a homozygous line is comparatively easy rather than the bialistic. And so if there is a low copy, 1 or 2 copies, then we can expect the less pleiotropic effect. In case of the bialistic, for the more copies, plants show more pleiotropic effect. But for the bialistic, it has some advantage also. That in case of the agrobacterium, I have mentioned already that overgrowth, the agrobacterium overgrowth, if the washing is not proper, then it can kill, it can hamper the explant growth or the regeneration. Not only this, in the next generation, T2, T3, we can get some seed or the propagules, which also contain the agrobacterium strain. And these can generate the false positive in the transgenic. So when we screen through genomic DNA PCR or the gene-specific priming. So, but in case of the bialistic, there is no question of agrobacterium overgrowth. There is no question of this type of false positive due to the agrobacterium stain or due to the agrobacterium infection. And another advantage of the bialistic is that if we need to develop a plant from the plastid, then only option is the bialistic. Through bialistic, we can generate the transformed plant from the plastid. The generation of a transformed plant from plastid is called the transplastomic plant. And these transplastomic plants are essential where we maintain the cytoplasmic sterility, where we generate a plant or the character which will present mainly toward the female generation or female plant. That character will pass through the female character. From there, we can get a plant from the plastid transformation. So plastid transformation is also important, or transplastic plants is also important. And suppose in case of some genes, we need to transform it into the plastid because we need to express it in the plastid so that we only transform the plastid. So for that, bialistic transformation is only the option. So now this is the basis of overall picture of this gene transfer method to agrobacterium, whatever it may be, or to bialistic. First, we will clone the gene, then confirm that yes, that this is my PCR product, and then we'll construct the vector. This is a different kind of system of cloning, that is a gateway system of cloning. Don't worry about it because there are so many systems. Just follow the protocol, and we can definitely you will do that, because there are so many systems, gateway-based cloning system, Gibson cloning system, so many. But suppose through whatever you learned through TA or the rest standardization and based cloning, you have cloned your gene into the vector, and from this, this is the destination vector, pDNA carrying region, the binary vector. Here you have cloned your gene. This promoter you can change depending on your criteria, depending the plant's criteria, and then ultimately this binary vector will be introduced into the agrobacterium, and or through bialistic you can also transform it, and then the generation of the transformed plants through tissue culture and regeneration system, and then the selection of the regenerated plants basis of the different antibiotic selection media, different antibiotic selection like hygromycin, kanamycin, or the VASTA. VASTA is not the antibiotic, but VASTA is one type of selective marker. It can inhibit the amino acid biosynthesis pathway, mainly the aromatic amino acid. Anyways, and those plants which are regenerated or and the selected, those plants are grown under greenhouse condition for further analysis. So before that, before the other analysis like expression analysis or the metabolic profile, whatever it may be, any type of phenotypic analysis, we should confirm the presence of our desirable gene in this plant through genomic analysis. So next is the analysis of the plant. That part I will cover in my next lecture. How to characterize that, how to confirm, and then how to characterize. Characterization means the expression. First will be the expression. That I will show you in my next class. Okay. Thank you all.